It's a long and complicated story for me. So I actually trained as an analytical chemist in Germany. And early in my undergrad, actually got interested in more biological sciences and actually did a summer student internship in a plant biology lab. And then as I decided to do grad studies, I again started in analytical chemistry, and luckily my lab actually had a collaboration with a vascular biology lab in New York, and I was actually able to move to New York for 2.5 years and do research there as part of my PhD. Moved back to Germany, completed my PhD, then again went out in the big wide world, actually moved to Toronto to do my postdoc in proteomics, which is then really combining my interests of analytical chemistry and biology, and eventually got stuck here in Toronto. Plant biology really was for me into kind of biological applications of analytical chemistry, and that kind of sparked my interest in biological research, versus most classic analytical chemists work on very technical applications, and I was always interested in combining these two skills, which proteomics really is actually the absolute perfect fusion of this. Yeah, that's also, I was actually always somebody that never planned ahead that far. I was always kind of stuck in the moment. I always wanted to do the best thing in that lab at that project that I was working in. I was in Toronto as a postdoc in proteomics when proteomics just became big, working on kind of mass spec application to disease mouse models. And as I then was completing my postdoc and interviewed, all over the world, Toronto for me was kind of the best offer I got. And as I started at Princess Margaret Cancer Center, more and more slid into kind of cancer research. I'm a very techie guy when I started, right? So you could apply this to pretty much anything. When I started my lab, actually probably only 40–50% of it was cancer research. Now it's 100% really. Yeah, for sure. Obviously Toronto is a spectacular, scientific community. And for me, it was probably the most kind of direct and easy way into an independent research career, because I already had a lot of collaborators here in Toronto. So it was actually somewhat easier setting my lab up rather than going somewhere else and starting completely fresh. Actually helped me quite a bit, when I was a junior PI. Having some senior colleagues that I already worked with as a postdoc kind of advising me. As I already said, I'm a mass spec, or my lab is a mass spec based proteomics group, and we have various areas of interest spanning technology development to more translational cancer proteomics to some very basic cell biological applications of mass spectrometry. So here that's basically one of the mass spectrometers, so it's liquid chromatography mass spectrometry. So here we're separating peptides from whatever samples we're interested in, be it urines or plasma membranes or very small tissue pieces. And we separate digested peptides by hydrophobicity and then identify and quantify them by mass spectrometry using various different ways of operating these instruments. I'm particularly interested in developing tools to quantitatively map out the cell surface proteome. And then, and that's actually where mass spec really is only the starting point. So it's maybe half a year of mass spec and 5 years of wet lab biology then. So we kind of span this whole spectrum, right, from mass spec to computational biology to somewhat wet lab, classic cell biology. So everybody in my lab kind of needs to be able to speak all these languages, right? So all students have to do their own mass spectrometry. There's technicians and maybe more senior postdocs that can help them, but it's not just handing over a vial to somebody else and doing it. You have to learn this. You might not necessarily have to be an absolute expert in it, but you have to run your own samples and be responsible for it. Computational, that depends a little bit more. There I have a full-time person that helps in the analysis. Nevertheless, I do encourage my students to kind of pick up R and, you know, learn to understand and analyze their own data, and then wet lab again. I mean, it depends a little bit. If a project is more, you know, biomarker clinical proteomics, that would probably require more mass spec and computational analysis, not so much wet lab. If a project is more on surface marker identification, then it's kind of a 1/3, 1/3, 1/3. Right? I mean, it spans the whole spectrum. I would say the vast majority, probably where we're most innovative, is mass spectrometry. That's kind of our bread and butter. But I mean, we do, you know, animal models, you know, cell culture, patient tissues, some computational biology, even though there we're probably not pushing the borders. There we're using other people's tools and maybe tweaking and optimizing them. You can't play on all fronts. You have to somewhat focus yourself. So we're constantly trying to improve and optimize technologies for liquid biopsy or biomarker work. Most of our work has been in prostate cancer, in particular in some type of urines. They're called post-DAE urines. So that's voided urines following a digital rectal examination. It's urine that contains prosthetic secretions. So here what we're actually doing now, and that's very much driven by technology, new technology in mass spectrometry, where we're now able to analyze much smaller volumes. We used to require 4 milliliters of these fluids. Now it's 200 microliters. It used to take a day to analyze a sample. Now we can analyze 10 to 15 samples a day. So now that allows us to ask much bigger questions, where we can actually do discovery using hundreds of samples. So we're actually kind of powered now in asking some of the clinical questions that we're interested in. So that's one innovative field we're working on. And then what we're also very interested in, it's kind of in line with what the RNA-seq world has done, single-cell, you know, RNA-seq. So there's a kind of a push in proteomics. Can one do single-cell proteomics? So I'm not sure if that's possible, but we're interested in at least developing technologies or optimizing technologies. There's some papers in that field that are coming out now. So we're interested in adapting or improving on these reports on using extremely small input material. And preliminary data from the lab suggests you can actually get down to maybe 50 cells and get reasonably deep proteome coverage. Maybe 2,000-3,000 proteins quantified in these samples. Yeah. So we cannot amplify, so DNA, RNA you can amplify, and our biophysical properties are vastly different. You know, DNA, RNA biophysical properties are somewhat similar. You know, a cytosolic or a membrane protein or a very small or a very large protein, they're very different to handle them. You know, you need to kind of solubilize, extract, digest these molecules to be able to identify or quantify them in a mass spectrometer, and that's difficult, right? And then of course, you know, you have as you can, and then of course also the dynamic range in proteins is tremendous, right? The dynamic range and the complexity. So I mean, in any given cell, there's probably several 100,000 different variants of a protein, with 10 orders of magnitude in concentration range. So you can see it's a huge analytical chemistry problem. That's the challenge. I have many collaborators, and you know, this kind of enabled and drove my career over the last, you know, 15 years really, from postdoc to PI. I mean, when I started my lab, I think Jenny Rosand was actually very supportive and was a great collaborator. In particular, Brian Cox, a postdoc in her lab that is now actually faculty in the Department of Physiology at U of T. In the last couple of years, I would say Paul Boutros is a very close collaborator of mine, where we're actually very interested in, you know, combining genomics, transcriptomics, and proteomics, building the computational work around this. And then several clinicians, you know, Stanley Liu at Sunnybrook in our prostate cancer work, Fifi Liu in some of the head and neck cancer work. And then even some companies, so CDRD, so the Center for Drug Research and Development in Vancouver, we're collaborating closely with them on building potentially therapeutic antibodies for some of the surface targets we identified. So it's a tricky kind of topic within, our department. There's, some faculty. I think students mostly like the rotation program. Faculty is probably somewhat of a split. So, rotations are very common in US schools. They're completely uncommon in European schools, so the background I come from. Within Canada, it's maybe a split. Some have it, some don't. So I think they're actually now, since we had it at least in the biology stream of MBP for I think 6 or 7 years, I think they're actually very useful. They're very useful for both students and faculty in finding a perfect match. And that's extremely important, right? For you to find a good match. It's not all of it, but that's an important point for you to be successful. So rotation student, I've done various things. Nowadays, I give them little mini projects, right, where they can actually hopefully get some kind of results in those 5 weeks. And their project potentially can build on that. I've also done it where I put a student with a rotation student with a more senior person in the lab and, ask them to help out, right? For a potential student to come in, I think it's extremely important for a student to take initiative, look at our webpage, and maybe look at 5 or 6 labs that they potentially could be interested. It's extremely important to do this, take the initiative, and go and speak to these PIs. And then I think that rotation program really then gives you some data, right? Now you actually have data. Before you didn't. I never did a rotation. You go in a lab and that culture of that lab or the style of that PI or even the projects you actually gonna work on then may not be what you like. And then you were kind of stuck with this, right? Now it gives you, maybe 5 weeks is not enough to fully gauge this, but it gives you data, right? Is your colleagues in the lab, are they helpful? Can you get along with them? What's the style of the PI? What projects are actually going on in this lab? So I think you gotta go in with an open mind and really evaluate these labs in an honest way and then make an informed decision. And then you also got to stick with that decision, right? You got to take some responsibility. You can't say first experiment doesn't work, oh, I should have gone to the other lab. That's not productive, right? Well, for me, actually, attitude and work ethics is probably the most important thing, right? Are you good in a group? Are you somebody that can fit into a group? Are you willing to kind of work hard? As you know, in science, that's not like a job where you start at 9 and then you drop your pen at 5. That's just not the way it is, right? You're competing nowadays basically with every grad student on this planet, right? So you have to be willing, you have to like this, and if you like it, you have to be willing to kind of put the time in. That's all of us had to do this. So that's very important to me. And then it depends a little bit in what type of project I would place a student on. So if it's more of a mass spec tech project, I would prefer if that student has maybe an analytical chemistry or physical chemistry background with some experience in mass spectrometry. That's actually not so easy. There's have been few students that I had in the last 10 years that had that. I had some, but they're not so commonly applying to MBP. And if it's more of a wet lab project, then of course, some wet lab skills would of course be highly appreciated. So on one hand, I'm super kind of hands-on. Until a couple years ago, I actually still worked in the lab. Now I actually, unfortunately, don't have that time anymore. But I do meet with everybody one-on-one once a week. And if the project is kind of like at a critical stage, I probably speak to them every day. So it's not that I necessarily double check on them or look over their shoulders, just, I like to have this constant kind of feedback and discussion on projects. I would be very happy if they do it themselves or if I get involved. I mean, in general, actually, every student in my lab usually has more than one project, so that gives them a little bit of a balance. I usually want them to move them all, and then obviously, from time to time, one project becomes more promising or important, and put a little bit more weight on that, and put a little bit the second project on a back burner. And usually what I do is I give them one project where it's almost guaranteed to succeed, and one that's a bit more high risk, high reward. And every student that came out of my lab to date, so there's been 5 PhD students, they all had their several first author papers. But they should, otherwise you shouldn't be getting a PhD. And that's tough. I mean, that's kind of like where, me as a supervisor's job is kind of to see what the problem could be. Just by having more experience, we could then maybe find alternative routes, around that problem, or shift the focus of the project a little bit. So that's probably my job. But it is obviously, that's science. You also have to realize this, right? That probably 90% of what we do is a failure. And this 10% where it works, that gives you kind of the kick. You have to kind of learn this and learn to deal with this. That's also kind of, to my mind, part of training a student in becoming a scientist, that you have to be able to, on one hand, when you leave at night, let this somewhat go. On the other hand, also not let it go, right? I mean, you almost have to constantly think, what can I do better? What would somebody else do? Who's competing with me? So that was actually that's probably one of my strengths, right? That I can be kind of just like a bloodhound almost, right? Just not let it go, right? And just you bang your head against the wall until you break through. So that's something I think a student has to learn, and also accept that's something that will likely happen to you in science. We have lab meetings. The lab isn't so big. There are 7 to 10 people usually in my lab. So, and of those, I would say half of them are either postdocs or students, and some are kind of full-time staff that their job is really to support those, students or postdocs. So absolutely, they should know what their colleagues do, and they do. And I very much encourage that they actually help each other. It's, for me, actually something extremely important. Your lab member should be kind of your biggest support, right? I mean, if they're not your biggest support, the guy 5 floors down definitely will not be your biggest support. So it's quite important to build that culture. So it's, to my mind, actually an extremely important part of training, right? It's not just becoming the best experimenter. I mean, that's then almost like being a technician. Not that this is something bad, but you have to learn kind of to plan your project, find people that can help you, present it, write it. So all of this, is part of becoming a scientist. And I train everybody as if they would become an academic scientist. That's kind of the bar I have. Not forcing them to become an academic science, but that's kind of what I expect them to become, kind of like a rounded scientist. Yeah, every student that came through my lab, again, those 5, every single one of them has gone to several conferences. I would say every single one of them actually gave oral presentations at scientific talks. I encourage it. Obviously, this is always somewhat, dependent on funding, but there's lots of actually funding support for students to go to conferences. The only thing I require them is to have something they can present. So it should be at least a poster or a talk. Just going there and attending, that's maybe not, so in the first year, maybe not, but. Actually, all the students out of my lab that graduated so far, not a single one actually went into an academic career yet, right? Which is fine. I mean, look, you can look at statistics, for our department or for U of T, 15% of our graduate students go into an academic career, right? Doesn't mean we shouldn't be training them for that. Maybe we have to train them even better to improve this. But 85% of our students will likely go somewhere else. I totally support it. I mean, at the end of the day, my job is to train you as the best scientist you can be, and then you have to decide what you want to do. That's not insulting to me at all. If you don't want to become a scientist, that's fine, and then I'll totally support this. And if I have connections or have an opinion, then I'm more than happy to share this with my students. As a matter of fact, I'm trying to set up an alumni day for MBP, so that will hopefully come next year and will be annually, where we bring back MBP alumni that are now successful in different fields, academia to biotech to whatever, consulting. So there's actually, hopefully you guys can get feedback on different career paths from people that are competent in that field. I cannot give you any advice on what you do in a consulting job. I can give you advice what you need to do to become an academic scientist, and that advice will probably be extremely similar from every faculty that you speak to. Right? There's many. So on one hand, right, I represent the students and the faculty at the Faculty of Medicine, right? And I report back to the students and the faculty in our exec meeting, and that then hopefully gets, amplified to all the students and all the faculty members on certain policies, or recommendations that are being made at the Faculty of Medicine. So an example would be that you guys, I'm sure, interested would be the harmonized stipend agreement, right? There's all the chairs in the room, and we probably don't always agree on all of these things, but we try to negotiate these things, right? There's several, right? So I want to have a bit more transparency in the decisions that are made, and some of this we're already implementing. So for example, all decisions, obviously I'm responsible for it, but I'm not the only one that makes a decision. The grad coordinators, the administrative staff, and then there is a executive committee, and that's where decisions are made and approved. So the executive committee minutes are now made public, so people actually can read, what we're actually deciding there. So that, I think, is very important. I also want committee memberships somewhat more structured. You can't be on a major committee, for example, the Appointments and Promotions Committee, forever. You have a 5-year term that can be renewed once. Like with most, like a chair can only have 2 5-year terms, and that's good, right? You shouldn't be sitting there forever. You burn out. So that's number one. Number two, and again, the students, I think, somewhat requested or asked for this, is to have these alumni days to give the grad students some kind of different perspective on what other careers exist outside from academia, where I can be the most competent in advising you. But I think that's very important. And then the third, and that's probably the biggest and most difficult thing for me over the next 5 years, is to kind of brand MBP nationally and internationally a little bit better. So if there's a student within Canada that's interested in translational cancer research or in medical imaging, their first thought should be MBP, right? So that requires us to improve our webpage. We will hire a communications person that has been approved, so that advertisement is actually being sent out soon. And that person's job is to help us in branding the department better, social media and all these kind of things. MBP is one of 10 basic science departments within the Faculty of Medicine at U of T. And we are probably the most, translational, patient-oriented department within these 10, even though our name might not fully recapitulate this. So we're certainly leading in medical imaging and in cancer biology, be it basic to translational to clinical. That's what separates us from many of the other departments. Not that this wouldn't exist in any of them, but I think we're more focused on these 2 particular fields of research, medical imaging and cancer biology. There's other work going on in medical biophysics as well, regenerative medicine, stem cells. There's even some neurobiology and cardiovascular biology going on. But these first 2 themes, if you want to call it. They're really the core of our department. It came because the students complained about it. That's why we have a new modular system. I mean, there's other departments that have this already. So Molgen, for example, had a modular structure before us. Biochemistry also has a modular structure, and they're probably the most similar departments to us. And so we brought the modular structure into MBP. It gives actually students more flexibility in taking smaller courses that better fit their interest or their research topics that they're working on. But also keep in mind, right, medical biophysics, really a focus is on a thesis. So it's a thesis-based degree. The courses are really just to supplement this. And you know, some of you have taken these modules. They're a bit granular, right? So I mean, just by taking one module, you certainly not be an expert in this. So you are then expected to read around it if required. And that's being a scientist, for sure. Right? I wouldn't change so much of what I'm doing right now. I probably would just do it in a bigger scale even, right? So if I had unlimited funding, I mean, the first thing I would do, so mass spectrometry is like the Formula One, right? There's like new instruments coming out all the time. It would be great if we could always have, you know, the newest instruments, several of them. That would be great. And then for a lot of our clinical proteomics work with those newer instruments, you can actually run many, many more samples. So we could do, you know, liquid biomarker discovery, not on 100, but on 500 samples, right? You'd be just so much more powered to hopefully find signatures that ultimately impact, you know, clinical decision making. I mean, that's what it all comes down to, right? And then another thing I'm very interested, and we finished one smaller project recently with Paul Boutros' lab in collaboration, is combining proteomics with next-gen sequencing data sets, right? And there's some very interesting observations that come out in the context of tissue-based biomarkers. Combining DNA, RNA, protein measurements actually dramatically improve the performance of biomarkers. So this is actually, to my mind, the first evidence that these multi-omic markers really could potentially further improve the performance of a signature. I actually do. I mean, I've always been an extremely active person. I mean, I was, you know, when I was a kid, I was involved in competitive sports my whole life, and I, to this day, have a very hard time sitting still, as you can probably see on camera. So I still do actually quite a bit of sport. In the summer, I do road biking. If I have time, sometimes I play golf. Yeah, there's really, I mean, as a more traditional profession, I think that's actually the perfect job for me. I wouldn't actually want to do anything else. When I was in high school, I wanted to be a professional athlete, but then eventually I realized that probably won't happen. So that would have been probably the only other thing I would want it to do. But, you know, didn't happen. So for me, really, science was actually, once I put my mind to it, that really is for me actually the perfect job. I would have a very hard time working in an office setting, and so. Everybody in my lab and kind of every student now at MBP, you know, can just come by and see if my door is open, and then if I have time, I'm happy to talk to them. I mean, they can either send me an email or just come by the office, right? That's probably much easier. Coming by the office is probably the preferred way to do it, because we get so many emails, sometimes they slip by. So if I don't reply, just send it again.
Perfect. Thank you so much again, Dr. Kislinger, for answering all these amazing questions and taking your time to speak with us today.
Happy to do it. Thanks for interviewing me. You guys gonna regret this, filming me for this, I'll tell you that. Nobody will ever come to MBP.